discussing results obtained on oncogenic kinase mutations in circulating plasma DNA in a big randomized phase 3 study that compared regorafenib versus placebo in TKI-refractory metastatic GIST patients.
Thanks, Pepe. Let me close that. Yep. Perfect, thank you. So good afternoon. On behalf of the worldwide team, this is a bit of a technology talk about a disease that really came to define personalized therapy for a certain type of genomic abnormality. This gastrointestinal stromal tumor has been a poster child for a disease in which a single gene mutation drives the cancer. Either the signaling enzyme KIT or PDGFRA is mutated and uncontrollably activated. Targeted therapies like imatinib or sunitinib were developed by our team with worldwide collaborations really to inhibit these mutationally activated kinases and have dramatically improved patient outcomes, a disease which would have killed people within 6 months, metastatic GIST, before these tyrosine kinase inhibitors or TKIs were available. Patients on the median are living more than 5 years now, and about 20% of people are still alive 12 years later taking a tyrosine kinase inhibitor. However, what we've learned from this disease will inform other more common cancers, genomically complex cancers, just like head and neck cancer or lung cancer, because we've watched a series of new mutations evolving in GIST to cause resistance to the standard targeted therapies. And detecting these drug-resistant mutations can be important to help us develop new drugs and develop new curative combination therapies, and eventually, hopefully, to choose therapies with more precision for our patients. Let me give you a picture of this. Here's a patient with metastatic GIST in the liver. Baseline 1 mutation is detectable by traditional sequencing, a KIT exon 9 mutation. After 1 month on this TKI, first line imatinib, you see most of the tumor turns black. That's death and destruction inside the tumor and a myxoid degeneration. And if you wait now 9 months, you see those white spots in there, blow that tumor up, and you see those white spots are growing clones within one big tumor. This patient was nice enough to let us take that out and analyze those white spots. What we see here is the first mutation, the exon 9, and then resistance mutation number 1 in that corner of the tumor. In this corner of the tumor, we see the first mutation, exon 9, and a second mutation, and over here, yet a third resistance mutation, all in the same driver gene driving the tumor. So you can imagine what the problem is. If this is one tumor and you just sample one corner of the tumor, you'll get one answer. You can't very well sample every tumor in the patient's body. So that's a limitation of tumor biopsies, and this talk is about a possible new solution. We know that these tumor tissue biopsies can only give certain samples, and tumors are heterogeneous. There is no one cancer genome. There are many cancer genomes, even in an individual. And so tumor cells are constantly leaking DNA into the bloodstream. So a sophisticated technology to detect those mutant DNA strands in the bloodstream may be able to give us a comprehensive picture of all the mutations in any individual patient. So this technology, which came academically out of Bert Vogelstein's lab and then has been commercialized by a company in Germany known as Einostics. I have no connection to Einostics whatsoever. I'm not a consultant or anything, but it turns out the company with whom I did collaborate to develop this kinase inhibitor contracted with Einostics as the contract research organization so we could do correlative science on blood samples from our patients in this big study. In your handout, you'll see this nice picture from a clinical cancer research review on this technology where blood can be expanded with certain primers by PCR and then a faint signal of mutant DNA in the bloodstream detected by this really interesting digital technology that has exquisite sensitivity. You can detect one mutant bit of DNA amongst 10,000 normal alleles, normal bits of the gene floating around. So we wanted to study drug resistance mutations in the patients coming into this phase 3 study of a new kinase inhibitor. I'm not going to give you too much details about this. This was the study we ran that led to the FDA registration of this drug as the third drug to treat GIST. FDA in the US approved it in February 2013. Big impact on controlling the disease. The correlative science was aimed at this question. Could the variety of known resistance mutations in the driver oncogenes be detected by this technology in blood samples? And is there any type of resistant mutation that did not respond to this new drug, regorafenib? The data on this actually are quite interesting. I won't go through them in great detail. It's there. I'm happy to talk to anybody about them. The bottom line is this. This technology detected 12% of patients with the most common KIT mutation in the bloodstream, compared to analysis of the original tumor, which detected 43%. So it's not as good. Why is that? Because there are certain kind of mutations that this technology is not great at, and it wasn't customized to detect that. What we were really customizing the PCR primers to do was to detect these secondary resistance mutations. Here's where it got interesting. We detected the resistance mutations in the blood in about 50%, 40%, 47% of patients, whereas the tumor tissue only showed that in about 12% of patients. So we get a more comprehensive view of the resistance mechanisms in this. We also saw a very nice concordance between the plasma, the blood sampling, and the tissue sampling when we had same samples in the same patients. And what you see here is the green bars are situations where the blood showed the resistance mutations that we were not able to find in the analysis of the tissue samples. Now, we've done a lot of work with the Broad Institute and others deep sequencing original tissues. And if you're lucky enough to have flash frozen tissue, very high sophistication and deep sequencing can sometimes show these in small copy numbers. But very few patients have that. They have formalin-fixed pickled tumor. So the nice thing is that everybody's got blood. So this technology lent itself to detecting the resistance mutations. When we looked at the results of the mutation analysis with outcomes, it was actually very interesting. Virtually all the mutation analyses showed essentially the same benefits with this new drug, regorafenib. So we can say with some degree of confidence that there's no big resistance group based on mutation subset, which is helpful as we think about approving a new drug, moving it out into clinical practice. There's no group of patients who shouldn't at least try this drug, and that was one of the reasons for this correlative science. So in conclusion, again, this technology was readily useful to detect resistance mutations in the blood, this beaming technology. They were more detectable in the blood than in the tumor tissue. And interestingly, for the future of our field, the majority of the resistance mutations were localized in a certain part of the gene known as the activation loop domain, which is part of the kinase that none of the current drugs really hit very well, and it's a target for future drug development. There's a high concordance between the blood and the tissue, and we were able to show that the activity of the drug in the patient population nicely spanned all of the types of mutations, but there were too few patients in this study. Even though it was a big international study, still was only about 200 patients because GIST is a rare disease. And so we didn't have enough patients with rare mutations in these other drivers for any kind of meaningful comment on the activity there. But to me, the interesting thing about this was that this is a technology that's applicable to multiple different kinds of solid tumors, giving us a tool to get comprehensive mutation analysis. Thank you for your attention.
Thank you, George. This is a very interesting piece of news, exactly for the reason that George just mentioned. One is the technology is now, I think, ready for prime time, not only for GIST, but also for other tumors where the presence of specific mutations and secondary mutation may be, in fact, of importance to know, and you could, in fact, monitor easily the patient without having to take multiple serial biopsies, but just simply by taking blood. GIST, however, is a rare tumor type, but it is the tumor type where most of the TKIs have been tested successfully at the beginning. Imatinib was the first one, and that, in fact, made the field explode into what we have now, drugs that are exquisitely active in particular subsets of patients that have specific genetic abnormalities, mainly mutations. So imatinib was the first one, sunitinib was the second one, and regorafenib now is the third one for this rare tumor type. It's quite interesting to see the story. Regorafenib is also registered now for colorectal cancer. It has been registered in September last year for patients who were failing prior chemotherapy. So if I would have to add a few things here, I think that the beaming technology is something that probably is going to be applied more and more to study patients who are developing resistance in a relatively simple way. One thing that we will have to be careful, though, about is the fact that heterogeneity in these tumors is a reality. And so what you pick up in the blood may not tell you everything what is happening in the tumor. So we will have to do multiple studies, serial studies, and potentially also take biopsies from these patients at the same time points to understand really what is happening in the blood and what is happening in the tumor. And having said this, I'd like to invite